Welcome to USMLE Step 1 Biochemistry lectures. In this lecture, we will talk about zinc. We all know that the zinc exists as Zn plus 2 form. Remember, it is not involved in redox reactions. So, this zinc is redox inert. But where is the zinc? Where is zinc highly essential? It is essential in the formation of zinc finger motif. Alright. What is zinc finger motif? In order to understand zinc finger motif, you will have to see the structure of a zinc finger motif. Imagine a DNA as a rope, and this rope needs to be stabilized in order to make sure that the genes are switched off and switched on. Whenever the DNA is stable and flexible, the genes are only able to be on and able to be off. For the stability to be maintained, the zinc finger is helpful. So what is a zinc finger? It is a finger-shaped protein which holds the DNA. Remember, it does not directly hold the DNA, rather it holds the proteins that are holding the DNA. So it usually acts as a metallic clip that is helpful to maintain the correct shape of the DNA. What happened was, the scientists have discovered several ways of proteins that bind to the DNA. One of them is the zinc finger motif. It was the second DNA binding structure whose 3D atomic structure was worked out. Scientists studied a protein called as transcription factor 3A. And this transcription factor 3A, the job is to turn on the genes. And if we specifically talk about what gene the transcription factor 3A is turning on, it is the 5S ribosomal RNA gene. And the function of the 5S RNA gene is to make ribosomes, which is involved in protein synthesis. Remember, without zinc, the transcription factor 3A does not work. So, zinc is essential for the functioning of transcription factor 3A. But when the scientists have examined the transcription factor 3A, they found that the transcription factor 3A contains 9 zinc ions. Remember this. So 9 zinc ions are found in transcription factor 3A. Therefore, it has 9 zinc fingers. Alright. And each finger grabs a part of the DNA. And each zinc finger has got the same pattern. That is cysteine amino acids or cysteine histidine amino acids, which are all the same amino acids. Alright. Now, whenever you see the arrangement of the zinc finger, it is usually in this shape. There is a zinc and there is cysteine amino acids. There is a zinc and there is cysteine and histidine that holds the DNA together. Remember, not all the zinc fingers are the same. Instead, they can have a cysteine or cysteine histidine or cysteine as you can see in this diagram. Alright. Some of the important receptors that has this zinc finger motif are steroid hormone receptors, thyroid hormone receptors, or vitamin D receptors, or estrogen receptors, or androgen receptors, or glucocorticoid receptors. All these contain the zinc fingers. Remember, DNA is a spiral structure and DNA has got a stable structure but is a flexible structure. And for this stability to be maintained, it is the zinc fingers that is important. Remember, how are the zinc fingers attached? They all sit on the one part of the DNA. Only one side of the DNA has got zinc fingers. Alright. Now, DNA has got a major groove and also has got a minor groove. A major groove and a minor groove. This is important because the major grooves are important for the transcription factors to be attached. And these transcription factors, whenever they are attached, they make the gene on or they make the gene off. This is why the major grooves and the minor grooves are important. Alright. Coming back to the transcription factor 3A. Transcription factor 3A, which is important, which is for which the zinc is important for its function. One zinc finger can read only a small part of the DNA. And remember, approximately 5 DNA base pairs are recognized by each zinc finger. So, transcription factor 3A has got 9 fingers and it can recognize at least 45 DNA base pairs because each finger can recognize 5 DNA base pairs. So, transcription factor 3A has got 9 fingers, so it can recognize 45 DNA base pairs. Alright. This is why the zinc fingers are important. Now, what happens if a zinc finger is damaged? If the zinc finger is damaged, the genes are not recognized properly, so the hormone or the enzyme cannot work properly. That is why the zinc fingers are important. One clinical example where the examiner can ask a question is related to rickets. Whenever there is a mutation or whenever there is an abnormal zinc finger, what happens is the vitamin D receptor cannot work properly. Whenever the vitamin D receptor is not working properly, the vitamin D cannot act properly. And this leads to deficiency state of vitamin D, which can lead to rickets. This is one classic example where zinc fingers are helpful. Now, we talk about other functions of zinc. One is, we already have seen that the zinc is involved in maintaining the structure of the DNA. Next, zinc is involved in zinc motifs. And where is the zinc helpful? Zinc is helpful in enzymes such as carbonic anhydrase, a very important enzyme, alkaline phosphatase, alcohol dehydrogenase, and superoxide dismutase. This superoxide dismutase has also got manganese as well. Now, carbonic anhydrase is involved in the reaction CO2 H2O, which is usually helpful in the formation of H2CO3, and the reverse can also happen. This is important for acid-base balance. Now, alcohol dehydrogenase, which is helpful in the conversion of ethanol to acetaldehyde. This is important for alcohol metabolism. Now, alkaline phosphatase removes the phosphate group. Alkaline phosphatase removes the phosphate group, and superoxide dismutase is involved in superoxide reactions. Remember, zinc acts as a Lewis acid. What is Lewis acid? It can accept the electron pair. It can stabilize the negative charges. And it activates water. Activates water for the nucleophilic attack. This is all about zinc. Thank you very much for listening. I'll see you in the next lecture.